So, messenger RNA gets chopped up. What happens to these little pieces? Well, it turns out that we also have, not only do we have many enzymes which are used to reproduce and repair DNA, but we also have enzymes that look for little pieces of DNA. And if they find a little piece, and defining little piece is something I'm not prepared to do, but if they find a little piece of DNA like this, they chew it up to pieces right away. And that has led people to investigate what are called inhibitory RNAs. And what they do is or an iRNA. You find that there's mRNA, tRNA, rRNA, and now we've added iRNA, interfering RNA. So if you have someone who has a metabolic abnormality, and that abnormality is genetic, it's based on their DNA, and they produce too much of something, and it's going to kill them. For instance, let's think of an example. In hemochromatosis, the body takes in too much iron, and you get iron loading in your pancreas. The iron, if it gets above a certain level, will make you diabetic because it will kill off the pancreas cells. It also goes into the liver, and if untreated or untaken care of, the liver will start to develop cirrhosis and liver failure over many decades. But the idea is, if you take a small piece of RNA, and it matches, it's complementary to the messenger RNA that you want to inhibit, this inhibitory RNA piece, this little piece, will attach to that, and those enzymes that look for little pieces will recognize that and the one that it's bound to. So it will chew both up. So the idea is, if we can find a way, maybe using those liposomes, those lipid bilayers, using those liposomes, if we can put inhibitory RNA inside that and inject it into somebody and make it so it goes where it's supposed to go, and that's saying a lot, it can bind with those cells and deliver the inhibitory RNA. And in vitro, with cells, this has been proven to work marvelously. And so it's a very active area of research. If you go to PubMed or even Google and put in iRNA, you'll see that there's a huge number of articles.
I don't know if I can remember my whole question.
Oh. Write it down.
Yeah. There's a way that they can take your DNA now and forecast what's going to happen.
Oh, yeah.
So how does that come in?
Well, because of my rheumatoid arthritis, I paid 23andMe $300 and something to do what are called SNPs, single nucleotide polymorphisms.
Did your children do the same thing?
No.
Okay.
I've wanted to include them, but they don't want it done. Turns out I have no known genetic risk for rheumatoid arthritis. So maybe if more people have that kind of thing done, they'll discover some areas that haven't been discovered yet. Let's see.
So they can, let's go a little further.
Sure.
So they can find many different things of diseases, really?
Yeah. What they do is they put it on a small chip that contains little pieces of DNA. The little pieces represent the differences between people. And so all they have to do is look on the little, it's like a computer chip almost, or it is actually. They just read off whether or not you do or do not have that particular mutation at that particular location. And they do it for hundreds and thousands of different locations. So DNA replication can cause abnormalities. What are those abnormalities? Well, mutations, point mutations. You can also get duplications. You get those insertions, the transposons. You can have deletions, because if the DNA is all lined up and it's being repaired, what if one side kinks? For instance, what if this point here actually represents a long piece which is coiled on itself? Well, now when it comes up and is reading, it's going to read this one, and it's going to read that one, and it's going to miss this entire thing. So there will be a deletion in the messenger RNA. If it turns out that that's useful in some way, maybe the protein that results from this will have a different target. Maybe it will metabolize a different sugar in a way that's useful. If that's true, then this will be selected for by natural selection, and the piece that's off will disappear. That's a big jump. Not only does the DNA get repaired and duplicated and all those errors can occur, I just explained to you earlier that the messenger RNA gets some of it trimmed out. Turns out that proteins need specific structures for specific three-dimensional configurations. And it turns out that even after a protein is made, you've got the DNA, the messenger RNA has already been chopped up to get rid of the introns, the protein is made, and it goes out into the world, and it turns out there are other enzymes which will chew up that piece of protein and remove segments of the protein. So when you get here, all you have is a strand with all those R's attached to them. And I'm just alternating just because. So you have a long string, and it has to fold in on itself so that it can attack its target, because these are usually structural or enzymes. And it turns out that even after it's been gone through this whole thing, an enzyme may come along and chop out some section in the middle. Now it can fold into its proper configuration. However, folding of proteins is extraordinarily complex and is an active area of research. And the research that's being done involves what is known as the grid or the cloud. It basically takes as many computers as people will let them use to try and work on every single possible move at every single
nuance.
Yeah, every single radical on the protein. And that takes a lot of time. And it turns out that proteins don't just turn into a structure and then stay that way. They're in a warm environment. What is heat? Vibration.
Would you say there's a director behind all this?
No, I don't think there is, because I don't think that anybody would arrange it this way if they were directing it. I don't think they would have the introns and exons. I don't think they would chop up the messenger RNA, and I think the protein would come out in its final configuration.
More complicated or sophisticated.
Well, I don't think it's good planning, though. Good design. It's not a good design. Excuse me. So DNA can get defects. Messenger RNA can have defects. Proteins can have defects. We're lucky to be alive. We're lucky to be walking on 2 feet and be older than 10 years old.
That's why you say you don't think it's a good plan.
I don't think it's a good plan because of all the errors that can happen.
Yeah.
Okay, we talked about crossover. Let's go back to that for a minute. I'm pretty much done with debunking the central dogma of biology. It's still a good simplification. That DNA, RNA, protein, that is still a very good top-level simplification. You just have to realize that's not all there is. Okay, we talked about crossover. I'm going to just draw two chromosomes here. Okay. we're in meiosis. We're in a gamete cell, a cell that produces sperm or a cell that produces eggs. We have, excuse me, let me back up. We have a fertilized egg, and it's going to undergo meiosis to end up with, it's going to undergo mitosis to end up with multiple cells. You can have crossover occur in a given location. I didn't say that well, I apologize. Which would end up with the results looking like that. Well, if there are genes that sit in here, because the amount of crossover is usually random, the relationship between those genes can be determined. That is, the distance between them. Let's say you have one gene that you know about and you're following, and you can do tests on it and you can analyze it in the lab. Well, if you look at the genes that are right next door, that didn't come out well, you'll find that they tend to cross over together. So you have what's called genetic linkage. If gene A goes across, it's very likely that gene B and gene C will go across, will cross over as well. And what that means is if you can find a gene that is a label, for instance, if we found a gene that said diabetic susceptibility, there would be other genes next to it, and you have to take into account that that may just be a marker. It may be the other genes which are actually causing the abnormalities. You following me? You happen to know protein A. You don't really know B or C, but they're the ones which are actually the active players in that disease. So linkage, chromosomal linkage or genetic linkage, is very important in analyzing recombinations. Could have said that clearer. I mentioned duplication as one of the things that DNA can do, and duplication is very important. We have, 11 or 12 proteins in the clotting system. So when you cut yourself, a whole set of proteins and enzymes are activated to create a clot so you don't just bleed to death from a little tiny prick. Where did they come from? Well, if we look at them, they look very similar. So you have 11 or 12 proteins, enzymes, which are very similar, but they have different active sites. And the suggestion, the hypothesis is that they all came from one protein which got duplicated. And each time it got duplicated, the duplicate could evolve separately. So you have one functioning protein, protein A. It does its thing, whatever that may be. It gets duplicated into protein B. Since A is there and functioning normally, B can mutate and become dead, a fossil, or it can mutate and become functional. And we think that that duplication of a fully intact gene is relatively commonplace. Remember that the human genome, something like 90 to 95% of it is not genes, and we're now learning that a lot of that is are pieces of controlling, initiating factors, inhibiting factors, and so on. So you can end up with what are called DNA fossils. What does that mean? It's mutated to a point where it isn't functional, but it's still in the DNA. And that is very useful because there is an approximate random series of events which cause mutations. We're always exposed to cosmic rays and the toxins in the food we eat. So gene B can mutate without having any effect on the animal it inhabits because A is still functioning. So if, for instance, B turned its start codon into a stop codon, it would never get read. Well, now we have a piece of DNA which is just sitting there doing nothing. However, we know that mutations happen at a certain rate. So we can look at that protein, see how many mutations there are, and we can guess how far back that duplication occurred.